Let's start with proteins. Okay, so our proteins have different structures and or different functions in our body. So we have structure, catalyst, movement, transport, and hormones. So our structural proteins includes the collagen, keratin, yung mga nasa hair natin, bone, skin, and nails. When you say catalyst, no, they make every reaction in our living system faster. So, kung matandaan ninyo, no, or kung sinabi man sa anatomy and physiology, if we do not have any enzyme or catalyst, so aside from a catalyst, no, which takes up a form of enzyme in our body, um, our proteins can also function as movement. So we have the muscles, no, which is made up of different types of protein, actin and myosin. If you're going to zoom in on the muscle fibers, no, on your anatomy physiology, I think I've already discussed muscular system. You'll see two types of protein, which is myosin and actin. Okay. We also have a function of transport. So one major protein now that responsible to our body in transportation of oxygen to the lungs and carbon dioxide from our tissue to our lungs to be expelled, no? Through exhalation is our hemoglobin. So other protein transport molecules also is available in cell membrane, which I think you will discuss in your anatomy physiology or in your histology. So number five function of our protein includes hormones. So many hormones are proteins. No, among them are insulin, oxytocin, and human growth hormone. So if you're familiar knowing ating insulin, this is a release by our beta cell in our pancreas to to lessen the sugar, no, or the amount of glucose level in our blood to avoid diabetes. So if you have functional problem or deficiency of insulin, no, this may result to diabetes. So we also have your oxytocin, which is released by our posterior pituitary gland. So oxytocin actually is our love hormone, and there's a lot of commercially, no, created or created copy of oxytocin, no, and can be sold commercially. So itong oxytocin natin sometimes ito yung sinasabing nilalagay lang sa perfume kasi it attracts love daw, nor it attracts opposite sex. So oxytocin is a love hormone released, no, when the mother of a children is lactating or whenever after sexual intercourse. So ito yung oxytocin. So this can result to a female usually, no, to have an urge na yakapin sila. So, oxytocin po yung responsible doon. You also have your human growth hormone, which is of course responsible for the primary and secondary growth for both female and male. Okay? Okay. Number six is for protection. There's a lot of protective characteristic of our protein. So this one of the protective characteristic is blood clotting involves the protein fibrinogen. So the body use protein called antibodies to fight diseases. So you're going to look back on these topics on your immunology and blood clotting in your hematology. So since I think I don't know if you have been taking your PMTP2 already, but if you are going to ask between serum and plasma, you should know the difference between them, ano? Serum doesn't have a fibrinogen where plasma contains fibrinogen. And plasma can only be formed through different set of tubes, which is your EDTA and your sodium citrate, where your serum can be produced through your red top or the non-coagulant, no, non-additive tube or the gel separator. So remember, serum doesn't contain fibrinogen because they have already utilized the fibrinogen for uh, for the red blood cell to be clotted, no, or when you are separating your serum to your packed red blood cell. While in your plasma, the fibrinogen is not still used. That's why fibrinogen in your plasma, um, fibrinogen in your plasma is still present. Mhm. So we also have protective function for our immunity, which is the antibodies, which is a form of also protein. Okay. We also have your storage, no, casein in milk and ovalbumin in eggs stores nutrients for newborn infants and birds. So we also have ferritin. No, this is a protein in the liver that stores iron. Okay. And the last function is regulation. So this regulation, there are certain proteins not only controls the expression of genes, but also control when gene expression takes place. So I think this will be discussed in your molecular subjects or immunology as well. So we also have two types, of protein. We have fibrous and we have your globular protein. So proteins are composed, of different amino acid. So this is the building block. If in carbohydrate, the building block is monosaccharide. In protein, the building block is amino acid. So this is the simplest, form of your protein. So this is a compound that contains both an amino group and carboxyl group. So if you're going to go back on our organic chemistry, now you know that amino has a nitrogen group, shown here, and we have your carboxyl group with COO, no? If you're going to remember, it's the carboxylic acid derivative. So since we do not have hydrogen out here, similar to this one, COOH, no, we have we call it carboxyl group. So in a protein, no, you'll notice there are two charges, positive and negative charge, which indicate that it is called or it exhibits a characteristic we call zwitterion. So when we say zwitterion, it can have both positive and negative salt. Therefore, no, it can be called a salt. Okay, so let's read this. Alpha amino acid, or an amino acid in which the amino group is on the carbon adjacent to the carboxyl group. Okay, again, an amino acid in which the amino group is on the amino group is on the carbon adjacent to the carboxyl group. So this one is your carboxyl group, right? Let me know if you cannot see my arrow. This one is your carboxyl group. The carbon attached to it is this carbon. No? And on this adjacent carbon, the amino group is attached to it. Okay, so although alpha amino acids are commonly written in an ionized form, so meaning it doesn't have any charges, they are more properly written in a zwitterion form or the internal salt with charges. So you can see that they have both positive and negative charge. So if you're going to remember, no, our inorganic chemistry, we have this concept we call chirality. So when we say chirality, it's the handedness of things or handedness of your molecules. What when we say handedness, no, when you put your hands together and you act as if you are praying, okay, you'll notice that your left hand is a mirror image of your right hand. No, so that is what we call handedness. If you're going to put your hand on the mirror, no, if you're going to put your hand in front of the mirror, you'll notice that the mirror image that mirror is showing is similar to your left hand. Okay. When you put your right hand in front of the mirror image that the mirror is producing is similar to your left hand. So that is your handedness. Okay. Meaning to say, even if you rotate your hand from clockwise or counterclockwise, sorry, this will never be the same as the other hand. Okay, so that means they are two different entity. Right? They are two different materials. They do two different things. So similarly to our molecules, they also exhibit chirality or handedness, meaning they are mirror image of one another, and this exhibits that they are different from one another. So this mirror image is best shown with a projection we call Fischer projection. No? We have D-alanine, D for dextrorotatory, and L for levorotatory. So we call it D and L. Okay? One thing you'll notice, and if you can remember on our chirality, is you'll know, no, that they exhibit chirality when the stereocenter or the central carbon, have different atom or molecule attached to it. Okay? Remember. So this, if you can remember on how to write line angle, no, you'll see that at the middle of this cross, there's a carbon out here. Okay? And that carbon, we have four different four different atom and molecule attached to it. So this can exhibit chirality. Okay? But if this CH3 will be replaced with just H, no? Or you'll just add a double bond oxygen out here. No, it doesn't exhibit chirality because the stereocenter is connected to more than one similar atom. Remember, the main rule is that your central carbon or your stereocenter should be connected to four different atoms or molecule. Okay? In that way, it will exhibit a chirality or it will exhibit a handedness. Okay? So for this one, no, it depends on how many carbons you have. So if you're going to check this carbon, it is attached to both two oxygen. So this will not exhibit. This carbon cannot be a stereocenter or a carbon with four different molecules attached to it because it has two oxygen attached to it. And one oxygen is also having a double bond. So you'll notice it here. No, there's a double bond attached to the red one. Sorry. Okay. How about this carbon at the bottom? This is also not a stereocenter because it has three hydrogen, similar hydrogen attached to it. So it can never show handedness or chirality. So there's only one stereocenter in this molecule, alanine. No? So it can only show two. No? We have a formula for that. So 2 raised to n. N meaning the number of stereocenter. No? So if we have two, okay, if we have just one stereocenter on alanine, no, we're just going to compute no, how many stereocenters or how many different molecules it can produce by using the formula 2 raised to the nth power. Okay, so the nth power is the stereocenter. If we have only one stereocenter out here, then this is 2 raised to 1, which is equivalent to 2, which makes this equation or this example correct because we only have two different molecule produced from one single formula. Okay, let me know if that is clear. Okay, another thing. Okay, okay, another example is the glyceraldehyde. Okay, so if you're going to check how many carbon are present in this equation, we have one, then two, and then three. Okay, if we're going to check this carbon at the very top, it is connected with the hydrogen and it is double bonded with the oxygen. So I hope there's no further needed explanation on the double bond of oxygen. This is an aldehyde. So it is, you must know that the carbon and oxygen has two double bond attached to it. Okay, so the carbon at the very bottom, cannot be stereocenter. Why? Because it has two hydrogen attached to it, right? So the carbon at the middle, if you're going to check, we have this CHO, we have this OH, we have this CH2OH, and we have this H. So they have four different atom and molecule attached to it. So this one at the center is a stereocenter. Now remembering the formula 2 raised to n power, that means 2 raised to 1 is 2. This thus makes this example correct that they have two different molecule coming from the same formula that is showing one stereocenter. Okay, so we have D glyceraldehyde and L glyceraldehyde. Okay, now protein derived alpha amino acid has different side chains. No? We have nonpolar, we have polar, and we have acidic or basic chain. Okay. For our non-polar side chains, these are ionizable group. This is shown in the form present in the highest concentration of 7. Okay. So we have alanine. So you just have to remember all these non-polar side chains. So if you can just if you can just create a mnemonic out of this. I think there's a mnemonic for this, and you should memorize because sometimes in your exam you're given different amino acid, and you just have to identify if it is non-polar or polar or acidic or basic. So for this one, we have alanine, glycine, isoleucine, leucine, methionine, phenylalanine, proline, tryptophan, and valine. Sometimes in your exam also, you might be asked which nonpolar amino acid has a sulfur as its side chain. So the answer for that is methionine. Okay. So these are just some example. Some other example question might be, which among the nonpolar amino acid contains both the NH2 and carboxylic or the COO as its side chain, or sorry, which among the amino acid um, contains what else? Sorry, contains a benzene ring, or which among the following of the nonpolar side chains is aromatics? So the answer for that is phenylalanine, proline, proline is a cyclic, so it is not an aromatic, and tryptophan. Phenylalanine and tryptophan. Okay, so you should remember the structure, the name, the shortcut name, and the shorthanded name, letter name. Okay, next we have polar side chain. So this is at pH 7.0. So similar to your nonpolar, you should remember and memorize its structure, okay, its name, Okay, it's other name, no? Since if there's other name and also the shorthand writing for it. Okay. And the third classification is our acidic and basic side chains. We have aspartic acid, we have glutamic acid. Sometimes aspartic acid is known as your aspartate, and glutamic acid sometimes is known as your glutamate. Okay. So we also have your arginine, histidine, and lysine. As you may see, aspartic acid and glutamic acid are both acidic, and arginine, histidine, lysine, no, or your HAL. No, my mnemonics on this one is HAL. Now they are all basic side chains. Okay, so for 19 of the 20, the alpha amino acid is primary. So the proline is the secondary. Okay, with the exception of glycine, the alpha carbon of each is the stereo center. Now why? Let's look back on our glycine. As we may see, what's the number two? With the exception of glycine, the alpha carbon of each is the stereo center. So in glycine, do we have a stereo center? Hold carbon 1H. As you may see in your glycine, we have two hydrogen bonded to your central carbon. So this carbon cannot also be your stereo center as it is attached to two oxygen. And one of the oxygen there is double bonded to your carbon. So in our glycine, we do not have any stereo center. Okay. So, but most of them have their alpha carbon as their stereo center. So they all exhibit D and L um, conformation. So we also have your isoleucine and threonine containing the second stereo center, meaning that isoleucine and threonine does have four stereo um, stereocentric molecule. So isoleucine and threonine has four, no? Because if we have two stereocenter and we apply the formula 2 raised to n, or being n the number of stereocenter, that means 2 raised to 2 will be coming up with four different molecule, no? On our or will be coming up with four stereoisomers, or the correct term is stereoisomers. Okay, so next, we have ionization versus pH. So the net charge on an amino acid depends on the pH of the solution in which it is dissolved. So if we dissolve an amino acid in water, it will present in an aqueous solution as is zwitterion. Remember class, now as we have said earlier in this PowerPoint, zwitterion have both positive and negative charge particles. So whenever we add amino acid in water, it shows its positive and negative charged particle, making it a salt. No? If we add a strong acid such as hydrochloric acid to bring the pH of the solution to acidic, no, or zero point 0.0, the strong acid donates a proton to the COO of the zwitterion, turning it into a positive ion, and vice versa to our acidic or to our basic. No, if you're going to add, say for example, NaOH. So what happened actually here is um okay, so what happened here is similar to when we add H3O, no? As we have remembered, no, a proton means a hydrogen and can also be identified as H3O or hydronium. So sometimes they can be interchangeable and sometimes called just proton. No, when HCl is dissociated in a water, it will become just hydrogen and chloride, correct? That hydrogen will be added to the electronegative oxygen attached to this amino acid. So what will happen is that the negativity will be removed because it will be replaced with just one hydrogen. Okay. And then what will remain is the NH3, so the positive charge. Okay. So that is what happened when you add an acidic molecule to it. When you add basic, no, a basic solution, or when you add this amino acid into a basic solution, what will happen is that one hydrogen from your NH3 or your amino group will donate its hydrogen to the OH of the basic substance. So what will happen is that this plus sign will be removed, making it a negatively charged molecule. Okay, I think we have an example for that. Okay. So this is what happened when you add the amino acid into a strong base like sodium hydroxide, no? In a sodium hydroxide dissociation, it will just become sodium and hydroxide. So this hydroxide, no, is this part, no? The hydroxide here is coming from your NaOH. What will happen is that this NH3 will donate its hydrogen, will donate its hydrogen to your OH, thus it will become H2O, no? And then your NH2 will be lacking one hydrogen, thus removing or emitting its positively charged. Okay, so to summarize, these reactions that we have is a reversible reaction that we can done repeatedly. Okay, um, this this reversible arrow, no, or double-headed arrow, is an indicatory that it can be done repeatedly, without causing any changes to the molecule itself. So we also have your isoelectric point. So these are the following for nonpolar and polar changes. Okay. For our cysteine, there are just some amino acid that we have to focus also. No? On our cysteine, we have SH or the mercaptyl or the sulfhydryl group attached to it, no? Which can be easily oxidized and become disulfide bonding. No? So remember, one classic example of your disulfide bond is the hair, especially those with kinky hair or curly hair. They have more disulfide bonding. So say for example, you go to a salon and you get your hair fixed, no? Through heat and what else? Chemical, right? They apply chemical. And what happened to your kinky and curly hair? It becomes straightened. Why? Because the disulfide bond is being broken, no? While on this process of heat and intense chemical addition of chemicals. So we'll discuss that later, no? That different ways for a protein or an amino acid to be denatured. The term used there is denatured. So let's focus first on our cysteine. Our cysteine, no, has an SH group or our sulfhydryl group, which can be, through oxidation, can become a disulfide bond. But remember, no, this is a reversible, indicated by double-headed arrow. This is a reversible reaction. So if we do reduction with different catalyst metal, no, this disulfide bond can in return become again a sulfhydryl group. Okay? So we have phenylalanine, tryptophan, and tyrosine. So these three contains the common thing that you'll notice on them, they contains aromatic ring. So they have aromatic ring for their side chain. So some very important amino acid is our tryptophan, which is a precursor of your neurotransmitter serotonin. So if you're going to check, serotonin can be induced by a lot of things, no? And a high or low level of serotonin can make a human being have a problem, no, of their hormones, and as well as in their way of thinking. Some other diseases and mental illness is a result of very low or very high serotonin level. Serotonin is very high in foods such as bananas and chocolate. No? Serotonin sometimes can make you happy or it can sometimes lift your mood. Okay? So the precursor of serotonin is your tryptophan, no? An amino acid we call tryptophan. Okay. Next, we have phenylalanine and tyrosines are precursor of your norepinephrine or epinephrine. No, this is more commonly known as your adrenaline or noradrenaline. So, as you may remember, no, in your anatomy physiology, these are produced by your adrenal medulla, norepinephrine and epinephrine. And they are the ones responsible for the fight or flight response and your rest and digest response. Okay. So from phenylalanine, it can become tyrosine, and through more further oxidation, it can become L-dopa, and then it becomes dopamine, which is a happy hormone, and your epinephrine, which is your adrenaline. Okay, so these are some hydroxylation process of your proline, lysine, and tyrosine examples. Okay, so let's move on to our peptides. So peptides is a more than one combination of your amino acid. So according to Emil Fischer, um, during that time in 1902, he proposed that proteins are long chains of amino acid joined by an amide bonds. No? This amide bonds we call now peptide bonds, no? Or the linkage is called peptide linkage. On our carbohydrate, we call it carbohydrate is glycosidic bond. Now, in our protein, we call it peptide bond. Okay? So the special name given to the amide bond between the alpha carboxyl group of one amino acid and an alpha group of another. So if we combine alanine and serine, now the combination of your carboxylic or negative group and your NH3 group, what will happen on this end, no? Okay, what will happen is that your oxygen here will be removed. Okay? And your two hydrogen atom from your amino group will also be removed. So this oxygen plus two hydrogen will form water. Okay? And what will happen? This bond will still need another bond, no? Because it's lacking. So it will be very highly reactive. So in that form, this bond will combine to your nitrogen that has only one remaining hydrogen. Okay? That bond formed from a carboxylic acid with your amino acid, with your amino group, no, is what we call the peptide bond. Therefore, we call this newly formed molecule alanyl serine, no? Or sometimes we call it shorthandedly with ala dash ser or serine. Okay? Okay. So we have peptide. So again, peptide is a short polymer of your amino acid. We have dipeptide from its name, di meaning two amino acid. We have tripeptide from its name, meaning three amino acid. Okay. And we also have your polypeptide, which is a macromolecule containing many amino acid. Now, we have how many polypeptides or how many amino acid before we considered it as a protein? So the answer to that is 30 to 50 amino acids should be joined by a peptide bond, enable for you to call it a macromolecule or what we call a protein. The individual amino acid units are sometimes called residues, no, instead of just amino acid. Okay? So we have a peptide bond. No? A peptide bond is typically written as a carbonyl bonded to an NH group, according to Linus Pauling. No, very familiar. Linus Pauling is also responsible for your electron configuration movement, or one of the three rules, in writing the electron movement. So according to Linus Pauling, however, he discovered that there is about 40% double bond character to the CN bond or carbonitrogen bond, and that the peptide bond between two amino acids is planar, no, in angle, which Pauling explained using the concept of resonance. Okay, so what does resonance mean? So if you're going to go back to your organic chem, no, resonance is best shown by our aromatics or by our benzene ring. That means it is a movement of an electron from one atom to another through through a form of a free electron pair to a double bond. No? For as long as there are more than one atom that has an electron unshared pair and a double bond in the molecule. So say for example in this part, your nitrogen has a one unshared pair of electrons. So that unshared pair of electron can be moved, you know, that unshared pair of electron can be moved and connected to your carbon in exchange, no? Since carbon can only cater to four bonding, no? If we create another bond to your carbon, we should move the double bond here from our carbon to our oxygen. Since ang oxygen natin can have more than two pair of unshared electron. So what will happen is that it would create a double bond and therefore the oxygen will have more than three. So it is just it's just portraying that this is just a movement of electron and it's moving a lot. No? Sometimes we just see a peptide bond, but if we are going to zoom no in these types of molecule they are moving reversibly, so from this form it can be on this form and from this form it can be on this form. So that means that according to Linus Pauling, 40% double bond character to the CN bond and the peptide bond between two amino acids is planar. So there are 40% of the double bond in the CN bond. Okay, so this one creates a 40% of the population of this type of amino acid. And then the 60% one is in the double bond on the CO, connection. You understand that? So in resonance, say for example, in our benzene ring, no? We call it delocalization pala. Sorry. I think these are a bit different, but the concept is still the same. In our benzene ring, now there are electron delocalization inside that pi look alike what they call that, pi look alike shape. Okay, so it's called an electron delocalization, meaning that the electron inside that benzene ring is continuously moving from one double bond to another. So similar to this, no, peptide bond resonance. 40% is on the CN bond, and 60% is a double bond, showing a double bond in CO bond. But they're still the same. They're still the same compound. Um, they have still the same number of atoms. They have still the same atoms, no? They have still the same structure, but they have different double bonding position. But it's still the same, no? Sometimes one is most commonly occurring in nature and the other is not. So next. Next is on writing peptides. So by convention, peptides are written from left to right. Remember, from left to right, beginning with the free amino group and ending with the free carboxyl group. So we call it sometimes C-terminal or N-terminal. Say, for example, we have a long chain of 5 amino acid connecting each other with a peptide bond, no? We will count on the left with the N-terminal amino acid. We call it terminal for a reason because it is the last amino group found in this peptide. So we also have the C-terminal or the last carboxyl group. Okay. Okay. So this is an example of more than one amino acid. So these arrows, no, is pertaining to the peptide bond created from one amino acid to another. So our N-terminal residue can be found here on our free amino group. So this one is your N-terminal and this one is your C-terminal. So if we're going to write the name for this um, amino acid, we're going to start on the left N-terminal residue up until your C-terminal or carboxyl terminal. Okay? Protein behaves as a zwitterion. So similar to water, no? Water is also a zwitterion. Proteins have an isoelectric point or yung PI natin tinatawag. At its isoelectric point, the protein has no net charge. At any pH above its PI, it has a net negative charge. At any pH below its PI, it has a net positive charge. So hemoglobin, for example, has an almost equal number of acidic and basic side chains. So its PI is 6.8. Serum albumin has more acidic, so its PI is 4.9. So proteins are least soluble in water and their isoelectric points and can be precipitated from solution when pH is equivalent to Pi. Okay, we have 4 levels of amino acid. We have primary structure, so these are the linear structure. We have the secondary structure that is divided into two. We have the alpha helix or the coiled one, and we have the beta pleated sheets or the folded one. Okay, we also have the tertiary structure or the 3D or three-dimensional structure of your polypeptide. And we have your quaternary structure, which is a spatial relationship and interaction between subunits in a protein that has more than one polypeptide chain. So primary structure in the sequence of amino acid in a polypeptide. So say for example, we can see that there are the number of peptides possible from the 20 protein derived amino acid is innermost. So how many this primary structure actually is the linear one. So you can write them for as long as you can, for as long as it can. So say for example, we have 20 peptides. How many combination can we create from that 20, sorry, say for example, we have 20 amino acid. How many combination can we create from the 20 amino acid? There's a lot. So multiply it by 20, we'll obtain 400 dipeptide possible, no? If we have 20 times 20 times 20, we have 8,000 tripeptide possible. The number of peptide possibly for a chain of n amino acid is 20 raised to n. So for a small protein of 60 amino acid, the number of possible protein that that can be created from the 60 amino acid is 10 raised to 78. Imagine how many molecules are there inside our body for a single collagen that we have in our body. So that's a huge number, no? Okay, so this is an example of your primary structures. I was saying, no, it is more like a linear structure of your protein. So if you're going to check, no, they're just straight. And sometimes when there is a sulfhydryl bond, no, you can see it. They are connected by another. So if we have, say, for example, we have 20 side chain like this one, and we have another 20 side chain as this one. So there's a lot of possible, ano, if we're going to remix what it has, no, or say, for example, we have to remix this part, or if you're going to connect this glycine to aspartate or glutamate, no, we will create another protein from that. So this example, no, na lang is the hormone insulin consists of the two polypeptide chain. We have A and B held together by a two disulfide bond. Okay. And the sequence shown here is the bovine insulin. So bovine is from a cow. So this is an insulin from a cow, but they are mostly used by commercially, no, for humans. Of course, we cannot extract insulin from a real human. So the insulin that being used by most of the diabetic patient came from a cow. Okay, so how important is the exact amino acid sequence? So human insulin is consist of two polypeptide chain, okay? Having a total of 51 amino acid, the two chains are connected by two interchain disulfide bonds. So let's look at this different insulin example. In human, though, at the chain 8, 9, and 10, we have threonine, serine, and isoleucine, okay? They are connected with the B chain, position 30, which is threonine. Or we have different on our B chain. Remember, this is the structure of our primary structure for bovine insulin. Okay? For cow, let's start for cow since what I have shown you before is a bovine insulin. We have alanine, serine, and valine at 8, 9, 10. Let's look at that. Okay, where is that? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this one is this part, alanine, serine, and valine. Okay, this is what's differentiating this insulin from human and other creatures. So alanine, serine, and valine. Okay, if we're going to replace that with threonine, serine, and isoleucine, no, on that chain, and the B chain will be replaced with threonine, that will be the human insulin. So that's the importance of the sequence that on the specific level of 8, 9, 10, alanine, serine, and valine can be replaced with any other amino acid, and then it will not make sense. No, this will not be insulin anymore if this will be replaced with any other amino acid not given in this table. Okay? So that's the importance of that sequence. On our B chain position number 30, the cow has alanine. Let's check. No, where is that? This one. This is the number 30. The cow or the bovine insulin has alanine. No? If we're going to replace that with threonine, no, with respectively, this one will also be changed. It will be a human insulin already. So that's the importance of different sequences. Just three parts, no, three or more, even one, no, one changes can make a total difference from what you are seeing as a sequence. Okay? So imagine kung and na-imagine niyo ba on how amazing the engineering of the human body is. One single change in that sequence will create a different molecule. And sometimes that molecule will not make sense at all or not function as it was before, as it should be. No? So that's just fascinating, no? How our blueprint was created. Okay, next, we also have your vasopressin and oxytocin. They are both non-peptides, meaning nine peptides, but have quite different biological function. Okay, so vasopressin is an antidiuretic, meaning they can induce urination, no? They can prevent urination. And oxytocin is, as I was saying, this was a a muscle contract. It can contract muscles. No, whenever a baby is being born, no, it can cause the uterus to expand more or to contract. Sorry, to contract. So the muscle of the breast, no, which aids in milk secretion, is also aided with oxytocin. So that's why I'm saying that whenever you are breastfeeding, no, oxytocin hormone is also released on your posterior pituitary gland. So there are just two hormone that is released on your posterior pituitary. We have vasopressin and oxytocin. And though they are both non-peptide, they have different biological functions. Okay, so this is an example of your vasopressin and your oxytocin. On your vasopressin, they are just different on your amino acid number 7 and number 2. And imagine they're just one to two changes in amino acid, and they have completely different biological function. Okay, so that's just fascinating. Okay, next number two is our secondary structure. As I was saying, we have two different secondary structure of your amino acid. We have alpha helix, which are coils or spirals, and we have a beta pleated shape. This one is an example of your alpha helix. So this is a bit complicated than your primary, which is just linear. Now it is a coil, no? And on this coil, there can be different amino acid combinations also. Okay, so we all have the C terminal and N terminal at the end of that coil. So we also have this section explaining your alpha helix. We have 3.6 amino acid per turn of that coil, and also the six atoms of each peptide bond lie in the same. So in this portrayal or in this demonstration, the C double bond group of peptide point in the opposite direction, although roughly parallel to the axis of the helix, and C double bond O group of each peptide is hydrogen bonded to NH. Okay. Next is the model is an alpha helix section of a polyalanine. So this is a combination of multiple number of alanine amino acid derived entirely from alanine. So this intrachain can create a cyclic or a spiral, no, formation, which is your alpha helix. This is an example of your beta pleated sheet. So as I was saying, this is like a folded paper. No? It has a N terminal and C terminal also, but you can see it's more complicated than your alpha helix. Okay. So on your beta pleated sheet, the six atom of each peptide bond of six pleated sheet lie in the same plane. Okay, and the rest are how they are being portrayed and being written in a 3D molecular space. So this is a random coil, no? This is a random coil of a protein. So this is what it looks like in real life. But if you're going to zoom it in, molecularly, you will notice that there are more than hundreds, no, of amino acid present in this random coil. Okay, many globular proteins contains all three kinds of the secondary in different parts of the molecules. We have alpha, beta, and a random coil next to a screen. So this one is a carboxypeptidase enzyme. No? So the beta-pleated sheet sections are shown in blue. So these blue ones are the folded sheet, and the alpha helix is in green, and the random coils are the orange strings. Okay, this is a collagen which has a triple helix. So this is made or structure of our skin. No? We also have a tertiary structure. So a tertiary structure is the 3D conformation, or you can see it as a not just a molecule. You can see it as a structure itself. So we have four ways that it is established. It's not linear. It's not in 2D, such as the coiling and such as the foldings. It was more than just that, a 3D molecular that has length, space, and you can see, no, on a 3D version of it. So I will show it later. Sorry. We have four ways that it can be stabilized. No, we have covalent bonds, especially for the cysteine side chains. We have hydrogen bonding, especially those amino acid with OH group. We have salt bridges for your amino and your carboxyl that both have positive and negative charge. And we have your hydrophobic interaction in case there are phenylalanine and isoleucine. So this is the stabilization of your tertiary structure. Okay. This one is your coil. This one is your folded sheet. These are connected by your metal ions. We have hydrophobic in case for our leucines, no, and our for our phenylalanine and isoleucines. We have disulfide bonding, and we have the electrostatic attraction, which is this one is salt bridges, and we have the hydrogen bonding. So if we're going to compress this one and creating a compact version of this one, you will see the tertiary structure or the 3D structure of this molecule. Okay. Next is we have the quaternary structure, which is the arrangement of polypeptide into covalently bonded aggregation. So one example of this tangible quaternary structure is the hemoglobin. Okay. It's not tangible by our hand, but if we're going to check in the microscope, the most accessible. It's the most accessible example that we can check. Actually, we cannot see hemoglobin. We can just see red blood cell. But if we're going to use molecular or transmission electron microscope, I think we can see hemoglobin as it is very small, no? We have two types of our hemoglobin. We have adult hemoglobin and fetal hemoglobin, and you can understand this more on your hematology. Okay? Adult hemoglobin has two alpha chains of 141 amino acid and beta chains of 146 amino acid. The difference is that on fetal hemoglobin present in your um, newborns, no, two alpha chains and two gamma chains are present. And the greatest thing is that fetal hemoglobin has greater affinity for oxygen as the newborn needs more oxygen than adult. Okay, since they have still developing lungs. So in adult hemoglobin, this fetal hemoglobin through time is being transformed into adult hemoglobin. So remember that red blood cell has just 120 days of living. So through that 120 days, no, they can change also what's inside of them, which is the hemoglobin. So each chain surrounds an iron containing heme unit. So this is an example of quaternary structure. This is hemoglobin. This pocket at the middle is where the oxygen is being carried. This one is the heme group, no, iron. And this is the alpha protein, and these are the beta amino acids. Okay, so this is a structure of heme. Okay, next we have integral membrane protein from quaternary structure. So let's see, these are integral membrane protein from rhodopsin. I think this is a part or a protein that can be found in your eyes. And we have also your outer mitochondrial membrane. This is an example of that with beta barrel and beta pleated sheath. Okay. On our, I think this is the last topic. We have denaturations. Okay, I'm sorry for the noises. For denaturations, the process of destroying the native conformation of protein by chemical or physical means. No, one example that I've given you a while ago is when we go to salon and we wanted to straighten our kinky or curly hair. The salon will apply heat and then will apply also chemicals. So in that form, heat and chemical destroy the disulfide bond responsible for making your hair kinky and curly. So some denaturating agent can be reversible, but some are not, no, or has permanent damage to our proteins. So our heat can be disrupt hydrogen bonding. So in globular proteins, it can cause unfolding of polypeptide chains, with the result of coagulation and precipitation may take place. Another example of this heat denaturation is when you fry an egg, no, the albumin in the egg or the white one, no, once a liquid portion, a viscous liquid, but when you fry it and when you apply heat to it, it coagulates or it become a solid form in the white form, no, nagkakaroon ng coagulation or pamumuo, no, the solidification of that viscous liquid, no. Sometimes it precipitates, no, on some other examples, but on your eggs it coagulates. Okay. So some denaturing agent, this one are chemicals. So we have 6 molarity aqueous urea. It can disrupt hydrogen bonding. We have surface active agent. So this is the STS or the sodium dodecyl benzene sulfonate or sulfate, sorry. It destroys hydrogen bonding also. So, detergent it's not that good, no, in our skin. That's why nagda-dry yung ating skin whenever we use it when washing our clothes because they are destroying the hydrogen bonding present on our skin. Our skin is made up of collagens and a lot of proteins, no, structural proteins. So when using detergents, no, of course, nagkakaroon ng disruption of your hydrogen bonding scale, no? So reducing agent also is a markup to ethanol. It gives disulfide bond. So if you're going to check your shampoos and your some of the agents used in the salons to make your hair straight, no, it contains 2 mercup to ethanol. So if you're going to check your shampoo just now, no, go to your bathrooms, check your shampoo that you are using. You'll notice if your shampoo is a straightening shampoo, you'll notice that there is a mercup to ethanol to it. Okay. We also have your heavy metal ions, which is a transition metal ions such as lead, mercury, and cadmium form water insoluble salt with your sulfide group. Okay, and lastly, we have alcohol. So the good thing is that this denaturation of protein aids us in killing and penetrating the bacterial wall and kill them by coagulating their proteins and hindi na sila magta-thrive. So the most effective way to penetrate the bacteria is actually not ethanol, but 70% isopropyl alcohol. That's why it's the recommended alcohol to cleanse your hand, not 70% ethanol, no? I notice also that when we are using 90 or 70% ethanol, it's usually dries the hand even with moisturizer in it. So I advise you guys to use 70% isopropyl alcohol in your household also. Okay? So I think that's the end of our chapter 2. Let me know if you have other question, if you have any further classification. Thank you so much for listening to this discussion. Okay? Thank you.